All right, welcome everybody. Happy New Year to everyone here. We're so glad that you can come out and join us for our first event of the 2023 year, I guess. I know us academics, we operate on our July to June schedule, but it's a new year for us, our first event. I'm really excited to have with us today Seth Garren. He's a UConn Health alum. I know that there's several UConn people here. But I believe his story will be applicable to many of us who have been on this journey. Some housekeeping things before we get started. Feel free to ask questions at any time in the chat. I'll be moderating the discussion. I'm sure there will be parts throughout the talk where we can pause and we can answer some questions. Otherwise, there will be plenty of time for questions at the end. We do ask that you leave your mics muted during the event. We are recording this seminar to be posted on our YouTube channel. But yeah, feel free to ask questions through the chat. And at the end, we can also unmute people if you're comfortable. Otherwise, I'll let Seth get started. Thank you for joining us today.
All right, thank you. Let me try taking over. All right, can everyone see my screen?
Yes.
All right, so I've decided to kind of take more of a autobiographical approach to my topic. I understand that the mission of this gathering and these types of seminars is to kind of provide information on potential career paths and what it's like to do those jobs. But for my purposes, I thought it would be helpful to kind of walk you through some of the less glamorous parts of the journey of academia and industry, as well as some of the life lessons that I learned in the process. And ultimately how kind of reflecting on some of the challenges and struggles and failures that I've had, and some of the unique opportunities that they provided, I think will bring some value and is worth sharing. So I have somewhat of a provocative title of how failures define my value, not suggesting that I'm a failure, but rather that they defined what types of value I will have the opportunity to create and how that can enable my career. So as was mentioned, I am an alumni from UConn Health. I had a somewhat unusually long tenure, starting my graduate program in 2007 and not graduating until 2015. So it's basically 8 years in grad school. And in addition to that, I had also done my undergraduate at the UConn Storrs campus. So functionally, I was at UConn for over a decade across both of those educational endeavors. And while it is a bit unusual for people to do the same graduate studies that they did their undergrad, the primary reason why I was interested in staying at UConn had to do with a lot of the major stem cell initiatives that the state was pursuing. And around 2007, stem cell was very heavily in the news because President George W. Bush made some announcements about restricting stem cell research for a lot of religious principles based around the sanctity of life and the ontology of embryonic humanity. But that left a lot of states to kind of pick up the funding. And so Connecticut very aggressively funded stem cell research and was kind of all over the news right as I was about to graduate in undergrad. So that definitely painted my interest in that as sort of being my next step. The other thing is that when I was in undergrad, I wasn't entirely sure what my path would be. I thought that, I knew I probably wanted to go into industry, but I wasn't sure exactly how much education would be appropriate. But I had learned that the PhD programs would pay you a modest but livable stipend. And then, if you decide that it's not for you, you can still opt to take a master's degree as part of like an exit alternative pathway. And so I decided, sure, why not? And then the question was which graduate program, and with all the news, this became apparent. And so as a result of that, I became very interested in trying to get into the lab of Dr. Renhei Xu, who had very recently joined the UConn Health, at the time called UConn Health Center, to start up a whole new stem cell core and to create some new stem cell lines. And this is very exciting because he actually came from the original lab of Dr. James Thompson, who was world famous for not only deriving the first human embryonic stem cells, but also discovering like one of the sets of reprogramming factors for induced pluripotent stem cells. And so as a result, he had come over and it was setting up a whole stem cell core facility and was working to derive novel stem cell lines as part of that initiative. So in 2008, I was given the opportunity to rotate in his lab and study the effects of transcription factors GATA1 and GATA2 on trophoblast differentiation, since it's sort of an easy and proximal cell type to kind of arrive at, and gave me a really good shot at culturing embryonic stem cells. But as things go, that you can run into some challenges when you're dealing with any lab, and I guess functionally that's the reason why we do rotations, is so that we can kind of not only get a sense of the topic, but also get a sense for lab fit and chemistry and how well you get along with others. And in my particular case, I had the too many cooks in a kitchen situation with my mentorship, where I kind of got caught in the middle of some procedural disagreements between the senior graduate student and the postdoc at the time, and that would sort of cause some stress and issues. And then additionally, the high profile nature of the lab sort of meant that the PI was under a lot of pressure to deliver new stem cell lines, and he didn't really have a lot of time or experience with mentorship. So at the end of my rotation, it became apparent that I really needed to kind of leave and search for another stem cell lab. So when I was looking through the list of other PIs who had taken on stem cell research, since that was still my topic of interest, I ended up joining Dr. Gordon Carmichael's lab, which had historically studied a lot of non-coding RNA regulatory mechanisms, specifically in polyomavirus. And he had also taken on some human embryonic stem cell grants to kind of expand that interest into the stem cell space. And so when I joined the lab, he had already had a senior graduate student there, and they were already on the stem cell grant. So while I was in a stem cell lab, I unfortunately didn't get to work directly with stem cells at that point. So I ended up working on his mouse polyomavirus grant, which was a very different biological system, but still had a lot of the same kind of genomic interrogations that they were specializing in. And so my thesis project ended up being focused on studying this tiny little virus. And it's somewhat of a remarkably small viral genome. It's a 5,000 base pair double-stranded DNA virus, and it only has 6 genes, but they're encoded on both strands in all possible reading frames. So from an evolutionary standpoint, it's about as efficient as you get, and it leans almost exclusively on the host for the rest of its processes. But that being said, this virus historically, which was discovered in like 1953, has actually been more or less the basis for a lot of the oncogenic signal transduction biology that we take for granted today, since it basically hijacks all of the canonical pathways that force a cell to divide. And that's basically what its early genes do. What Gordon's lab was particularly interested in was the sort of exotic ways in which this virus is regulating its RNA. In particular, it would rely on some sort of strange arrangements. You'd have 2 transcripts that are functioning in a collision course with one another between your early and late genes. So your early genes would code for your 3 different proteins that would more or less force the cell to divide and shut down some of the tumor suppressor signaling. And then the late genes would provide the viral coat proteins so that it could package up its progeny and ship them off to the next victim. And so the big question there is like how exactly does this regulation work? There were some kind of curious observations. So for example, the late strand would have this interesting effect of kind of doing a rolling circle transcription where we just kind of run around the entire genome a couple times. But then it would create this unique splicing pattern where it would have a little leader sequence that would kind of get spliced around, and it would correlate with the number of laps it happened to make before it stopped making RNA. So you create this sort of tandem leader repeat in the bottom right corner. And then another question that came up is you'd have these overlapping polyadenylation sites to sort of tell the transcription to stop. And if you were to remove that overlap, the late genes would fail to be generated. And so there was this kind of ongoing question of like, what is the relationship between late gene regulation, this polyadenylation overlap, and why is it necessary that cell division in particular is critical? If you inhibit cell division, you can't get late expression. If you remove the overlap, you can't get late expression. So what is that relationship? So one of the questions I ended up asking was, how can transcripts on a collision course produce double-stranded RNA? And part of the reason why we thought that the overlap was important is because it might produce double-stranded RNA that would be a target for adenosine deaminase acting on RNA, which is an enzyme that chemically modifies adenosines into inosines, which changes how the shape and their recognition by other enzymes and biological interactions. So that was ostensibly what I was supposed to be working on. So the strategy for this was I had this hypothesis that, well, first of all, if these are on a collision course with each other, we looked into like electron microscopy that's showing what happens if you put two promoters on a collision course, and they find that they just kind of stop. Like there's not really any room for them to pass each other as far as we could tell. So the hypothesis is, well, maybe it's like every other molecule is either early or late. But then the question is, why then does it need replication? Well, maybe replication creates little loci where you have lots of copies of it that could do either early or late, and they're right next to each other, which creates the proximity necessary for this RNA to hybridize and provide a target for ADAR. And so the idea here was to create a heterodimerization system where we would try to design these 4 different vectors and then use a sort of rapamycin analog to kind of force these artificial partial virus genomes to come into proximity to see if we could induce editing. Unfortunately, a lot of the cloning tools that we're using at the time, where you're basically just cutting and pasting bits of DNA into each other and then hoping you can shock a bacteria into taking it up, was really just extremely cumbersome. It was just taking way too long. And through just an immense amount of trial and error, I was already about 4 to 5 years into my PhD by the time we concluded the strategy wasn't going to work, and I basically had to start over from scratch. So that's the primary reason why it took me so long to graduate. Furthermore, the larger issues that I ran into was just a lot of psychological challenges, struggling with imposter syndrome, which I understand is relatively common in certain high-performing environments in academia. But I definitely had this strong sense that like everyone else seemed to know what they were doing. They were generating a lot more productive data. And I had an extremely difficult time getting guidance. In particular, the lab I was in kind of had a tradition of just letting the older graduate students guide the younger graduate students. But in my case, our senior graduate student was just wildly productive. Like she could just, pump out a paper a week. Every figure was beautiful, and she just didn't have any time to even show me where I could find a restriction enzyme, let alone like troubleshoot why my protocols aren't working. And so that just kind of created this vicious cycle of just feeling inadequate and inappropriate and like I just don't belong, and that it just wasn't happening. And the longer that went on, the kind of worse that got. The other issue I found is that I would frequently witness students being bullied by faculty. I would see a lot of cases where people would be kind of giving presentations and the faculty would just absolutely tear them to shreds. I didn't really understand why it needed to be so aggressive. And eventually one of the faculty ended up not getting tenure because they bullied students so badly that the students gave them a poor review and they were denied tenure. So like that's a common thing that can happen in academic circles where certain individuals might either believe in a tough love approach or they're just straight up bullying of them. As far as my own committee members were concerned, they were mostly they were either extremely harsh or very disinterested. So every time I would meet with them, I didn't feel like I was interacting with mentors or teachers. I felt like I was on like a Simon Cowell like show where I'm just constantly getting torn apart. And the biggest issue is I felt like naivete was treated like a fundamental character flaw. Like you are not allowed to not know something. If you don't know something, like you would just get chewed apart. And that just kind of shut down any attempt to ask clarifying questions. And then, of course, over time, this compounded. The longer my project was failing to produce results, the further I felt like I was falling behind. And this kind of just created a vicious cycle where I was terrified of asking for help and just feeling like an utter failure. So in the meantime, I just needed something else to do. So I decided to try looking outside to build confidence and leadership skills. So one of the other issues I had is that I was sort of lonely and wasn't having very much success connecting with people. So I tried turning to meetup.com, which I think a number of you might be familiar with, which is a platform where people can kind of just like utilize their existing infrastructure to advertise events. And so I decided like, well, I tried looking for events that had some of the topics I was interested in. You know, I'm a huge nerd. I like sci-fi, fantasy, anime, role-playing, etc. So I tried finding groups like that, but I noticed there weren't any. However, I did notice there were a lot of people in like a 50-mile radius who had those tags. So I figured, why don't I just try starting one of my own? So I started my own group. I called it the Geek Culture Group. And it ended up becoming wildly popular. Like lots of people showed up and we were able to do a wide variety of activities. And it was a really great way to kind of like both develop leadership skills and also make friends and kind of build my own community. And interestingly enough, it still exists to this day. Like on the top left, you can see we're playing a game I invented called Boffer Capture the Flag, which they still play every month at Elizabeth Park in Hartford. So if anybody happens to be interested, you can still search for it right now. So it's since been passed on to others, but they've been carrying it quite effectively. So ultimately what ended up happening is we ended up using NGS as a backup plan. Right around this time, Illumina sequencing had won out the sequencer race between all the different Celexa and Solid and other next-gen sequencers. Was starting to become more standardized, and the genetics and genome sciences department had kind of come on its own around 2012 to 2013. And at that time, NGS became a more viable approach. So ironically, my failure kind of let me bide my time until this new technology became an option. And once we pursued that, it became a really nice backup plan because, well, okay, I can't really get a good hypothesis story done in a reasonable amount of time. So why don't we just try doing an NGS data dump, because just the entire modality is novel and that'll just overwhelm the reviewers with data that we can kind of just get something out the door. So what I did is I took kind of a standard polyomavirus expression, but then we decided to focus it on both the virus and the host. And so by just doing some time courses with and without DNA replication inhibition, we're able to like kind of just do a global characterization of all the changes in a polyoma infection. And this showed, we're able to just basically kind of show all the hallmarks of what's happening and illustrate that you can kind of get this giant, like everything Google Earth view of what's happening in the infection and identify, the changes in the host. We can see the gene mutations that correspond with editing. We can see the late leader splicing events. We can see the changes in early and late expression. And it's kind of like, it's almost like a review paper off of a sequencer, so to speak. But it was enough to get published, which is how I was able to finally graduate. And so as I was finishing my PhD and getting ready to defend, I kind of found myself asking the seemingly stupid question, what exactly is a postdoc? Because, everybody seems to just naturally go from grad student to postdoc, but I never really thought about like what role the postdoc plays. Why do you get one? What's it necessary for? How do you get one? And of course, I assume everyone's familiar with the Piled High and Deep comics, which make fun of the academic experience, where they have a couple of different versions. Like when saying a postdoc is almost a requirement for an academic career these days. Employers want to see how well you perform as an independent researcher before they hire you. I don't really have that much independence. In theory, no, but given how much they'll ignore you, yes. Can I have that in writing? And so on and so forth. Or likewise, they'll just claim that postdoc just says, I have a PhD and not much else. And that's probably how they invented the title. So at the same time, one of my lab mates, as I was trying to ask about this and try to learn about that, had kind of introduced me to the idea of networking, because he was an MD PhD and he knew some people who were actually working in industry. And so he was able to give me warm introductions to them so that I could actually talk to them about their paths and specifically kind of get some firsthand experience from people who had different types of postdoc journeys towards industry. So I spoke with a person who had a postdoc before joining industry, a person who just joined industry straight from their PhD, and a person who did their postdoc in industry and kind of learned what each of the differences are. And it seemed to be basically that the people who jump straight from PhD to industry are the ones who just continue doing the very exact same thing they did in their PhD. People who did their postdoc before industry are ones that wanted to kind of expand their toolbox and experience. And the ones who do their postdocs in industry are the ones who will happen to answer some side question that's biologically interesting, but not necessarily directly related to a drug pipeline that a department might get funding for. And so I would end each interview by asking them for an additional warm introduction from their network so I could continue kind of going down the chain. And that's a fairly common networking practice. So then I decided, well, I guess I should find a postdoc. Well, this process was was a months-long process that kind of ended up with a lot of rejection. So, I kind of like the quote, sometimes you need a door slammed in your face before you can hear opportunity knock. As with anything, you're going to encounter a lot of rejection. I sent out countless applications. I went to multiple interviews. I was told I wasn't passionate enough about science. And so it was kind of a very demoralizing experience. But one day on a whim, I decided to send an application to a lab in Boston that seemed to be a lot more prestigious than all the others I was being rejected from. And then I kind of was confused because I ended up getting this single email response just asking me what's your citizenship status and when are you available to interview? And this is a little weird because every other time I'd send an application out to a postdoc, I get this long back and forth, detailed questions via email from the PI, just really closely scrutinizing everything. And then suddenly this group's just what's your citizenship status and your schedule? So okay. So I ended up interviewing at the Center for Systems Biology at Massachusetts General Hospital, which is where I ultimately got in and did my postdoc. And this is sort of the big opportunity I did not expect being able to qualify for. Here I was some grad student with basically zero confidence, failing a very tumultuous 8-year-long PhD, suddenly just jumping into the deep end of the big pond, so to speak. And so when I went there, I was very intimidated. And just the sort of terse and abrupt nature of the email made me think that this is going to be some sort of audition where I'm just sitting in a room with a bunch of nervous people who look like me. But in reality, it was the exact opposite. When I arrived there, everyone was super chill and laid back, and they just seemed to be relaxing and having fun. And the culture was just nothing I'd ever seen before. Nobody was taking anything seriously. They're all just kind of relaxed and confident and secure. And so I'm wow, this is interesting. So I ended up joining the lab of Dr. Ralph Weissletter at the Center for Systems Biology. So Dr. Ralph Weissletter is a fairly big name in the medical and biology community. He holds postings everywhere. So he's a professor at Harvard Medical School. He's a director for Center Systems Biology, and he's a attending clinician of interventional radiology at MGH, among a host of additional things and accolades. And his group primarily studies novel imaging methods for early detection of cancer, since he has a radiology background. But he also collaborates extensively with a number of biomedical engineers, and he has this massive multidisciplinary group. So he's almost like a super PI, where he kind of goes to Washington, comes back with millions and millions of dollars, and then he'll fund other PIs within his department. And so the department is almost like a super lab with a whole variety of different expertise. And so he'd have other PIs, postdocs, grad students, and they specialize in imaging, immunology, biomedical engineering, chemistry. And the reason why I ended up getting this postdoc is because he needed somebody with sequencing experience, because he had some questions about whole exome sequencing of the nucleic acid content of extracellular vesicles from pancreatic cancer patient plasma, which is a bit outside of his normal readout, since he's used to doing imaging readouts or flow cytometry readouts. So I kind of provided a new avenue of modality. So my initial project with him was to try to study liquid biopsies in pancreatic cancer. And so here the idea was that there were some reports that when people were trying to isolate these tiny little lipid-bound vesicles called exosomes. So a type of extracellular vesicles are 50 nanometers in diameter. And they're basically the cell's mailing system. They just kind of package stuff up and ship it out. It's a lot of proteins and RNA typically. But some people were finding DNA getting pulled down. And that DNA would also sometimes have detectable driver mutations, such as KRAS G12D. And so in that case, this was pursued as a potential avenue of liquid biopsy, where if you have a person who might have early stage pancreatic cancer and you could detect it by just looking at cell-free DNA or exosomal DNA, this might be a way to detect it earlier and potentially save their life, since one of the reasons pancreatic cancer is such a death sentence is you just don't know you have it until it's too late. And so one of the main questions we had is whether the DNA that's on the vesicles, are these tumors somehow literally shredding their genome, packaging it up and mailing it out in some weird gory effect, or is it somehow resulting from apoptosis, or is it just cell-free DNA getting stuck on the outside? And so throughout exploring this process and having like basically a year of very little progress, I eventually was able to get some electron microscopy support and was able to identify that it seemed to be the case that DNase treated exosomes no longer would have the DNA binding epitope. So it's probably just it's getting stuck on the outside, or we're just grabbing ctDNA. So I felt like this project wasn't really going anywhere, and I'm starting to get anxious because I'm like right back where I started. My project is just like, oh, I'm just kind of spinning my wheels, not getting anywhere, and I'm having all those same anxieties come up again. However, this time I decided to apply the lessons that I had learned from my mistakes in grad school. This time I was determined to only do one quick 2-year postdoc as a stepping stone and not just turn into a permadoc who's just sitting in academia in this sort of endless like rotation for 6 to 10 years that some people can do. And this time I decided to seek help. I sought help anywhere and everywhere I could find it. I would ask grad students, other postdocs, other PIs. I took elective classes in bioinformatics and clinical study design. I joined mentorship programs. I reached out to former UConn people. So some of my classmates who were in the area or other postdocs or other people who had done their postdoc when I did my PhD and then had moved on to their next places. I'd pester core facilities across multiple campuses, and just like anyone I could find, I would just ask. And then I had this really funny situation where like about a year into it, I like sat down with my PI and I'd put together this whole big presentation about why I didn't think the project was going to work and we should change projects. And he had apparently done the exact same thing. So here were both of us sitting on either side of this meeting, trying to aggressively persuade each other of the exact same thing. Like, and it was just funny watching us like awkwardly try to broach the subject with the exact same argument. So clearly we were on the same page. So again, what's the backup plan? Let's do a new sequencing technology. Well, if all else fails, let's just do a new sequencing technology. So if last time I did bulk sequencing as my backup plan, this time I did single cell sequencing as my backup plan. So at the time, 10x Genomics had come out with their first 3' single cell sequencing technology. And so for the second time, I learned a new omics technology as a backup for a failed experiment. But the other key difference is this time I got to work with a multidisciplinary team for the first time. And this included working with imaging and immunology experts. And we were able to publish a single cell story in under a year on the different cell types and states of tumor-associated macrophages. It actually came out to be a very beautiful story that ended up kind of foreshadowing some of the later work I would do in industry. So the basic gist of it is that there's this MC38 model that's a common popular tumor model in mice. It's basically a cell line derived from mouse colon cancer. You inject it under the skin and it grows into a tumor. And then you can treat that tumor with various treatments. The most popular treatment is called immune checkpoint blockade, where you take an antibody against the normal checkpoint signaling proteins like PD-1 or PD-L1 or CTLA, whatnot. In our case, we did PD-1. And that basically blocks the like, don't kill me signal that a lot of your cells will provide to immune cells that tumors tend to abuse when they're trying to avoid the immune system. And in the MC38 model, at least the PD-1 treatment is fairly successful. So it's a nice model for like what a best case scenario looks like for a checkpoint treatment. And so one of the questions that we had was like what effects do the tumor associated macrophages have in this setting and system? And so like naturally within the tumor microenvironment, there's a lot of actors. So you get like different macrophages, fibroblasts, et cetera, that are either for the tumor or against the tumor. You either have pro-inflammatory signals or you might have anti-inflammatory signals. And so there's this sort of very oversimplified duality to macrophages that the storytelling was focusing on, where there's M1 macrophages, which are more pro-inflammatory, and M2 macrophages, which are more anti-inflammatory. And so the question is like, what's going on with these M2 macrophages? Well, one of the markers for M2 macrophages is arginase 1, which is used to kind of mostly to deplete one of the metabolic precursors that's necessary for T cell function. And so we wanted to kind of dive into what are the different substates of macrophages. And so so what we did is we did single cell. We would take the cells, put them in the MC38 tumor model, remove the tumor, dissociate it, do a CD45 sort, and then single cell sequence all the results. And so for my part, I did the single cell sequencing, and then our microscopy experts would kind of do live intravital imaging, and they were able to show that there were two different types of macrophages that they could observe. One were these sort of rounder ones that would be around the periphery, ones that were more kind of elongated and stellate shaped that were in the center. And when I would look at the single cell, I'd also notice that there's this kind of continuum from younger to more differentiated. And so we put together this nice little story. We're able to just combine single cell and microscopy to kind of create a proto-spatial transcriptomic readout and show that it looks as if on the periphery, you get these rounder, younger macrophages, and then in the center, you get these older, more differentiated macrophages, and you can kind of show that through these different methods. And so that kind of gave you my ticket out to do other things. In the meantime, I was really able to take advantage of Boston itself. So the city provided many opportunities for attending networking events. So I joined a bunch of different groups. There was Biotech Tuesday, BioExchange, Mass Bio, and they'd have all these fun little events where you could just go to some random place. Sometimes I'd like go to this like rooftop bar where you're just kind of schmoozing with random people from all over and talking to people in industry and academia, and you know they have drinks and hors d'oeuvres. And I got a chance to meet a wide variety of people working in biotech and pharma and all that. And this allowed me to start building a network with a large number of what are called weak connections, which in networking terms are the people who you like barely know, but kind of have, because they're so distant, they can kind of open up entire new networks for you when you find some job posting that might have a connection to somebody. And things like that can increase the likelihood of you having a connection to a company with a job posting, or if you're looking for people to work for you further down the line, it can help you find future employees. And so at this point, I was trying to get into industry proper. One of the activities I did at the time is I joined like a little mentorship program where I got to talk to people who were currently in industry who would come back and mentor postdocs who were interested in going into industry. And it was like a nice little safe space for us to spend 90% of our time complaining about lab drama, but it also gave us an idea of like what other people's journeys look like. And it's kind of the spirit of the storytelling style I'm applying in this particular talk. And so ultimately, you know, I was putting out tons of applications and trying to, you know, find where I should fit. And ultimately I ended up doing 3 interviews. The first place I interviewed at was a place called Exosome Diagnostics. And I was primarily noticed that because the founder also happened to come from one of the professors in our group. Xandra Brakefield was his mentor. And since he came from her lab, I figured that was kind of a good weak networking connection to try to use as an angle to get in. So that at least landed me an interview, which went well until I gave them an answer to a question that kind of drained everyone's enthusiasm. And I think I had to do more with the fact that I just shared my honest opinion that I think protocols that are not easily reproducible are not good protocols. And so that kind of made them think like, yeah, this isn't going to work. It's like we need someone who just kind of bulldozes through like brute force library prep. Which in retrospect was probably not the best job, but like, you know, one they were considering. The next place I interviewed was Avello Biosciences. I interviewed there because I was mostly interested in their kind of novel strategy of using entire microbes as a drug. So there was other groups like Ceres that would kind of try to create artificial fecal transplants, but it's a very highly heterogeneous like product, whereas Avello is like, well, why don't we take one bug at a time and then just put it through the same drug testing pipeline. And they needed somebody to kind of build NGS, but they really needed somebody to build it from scratch. This would have been a very difficult role to start, and it would also be in a startup environment. So it was be kind of like an aggressive stretch situation. And then right as I was like planning to go to Avello, I got a response from Pfizer. Now Pfizer was funny because I had initially seen that they had a job posting a couple months ago. But when I tried applying for it, like I tried looking at the job posting, it like the link was dead, and the only place it showed up was this like third-party Mexican job site that clearly just didn't curate its dead links. But then it popped up again, and I guess what happened was the hiring manager had not yet refreshed it. This time I found it actually on Pfizer's core website, so I decided, sure, I'll apply, and they got back to me surprisingly quickly. And at the time, I was very focused on Avello, but I was like, sure, why not? I'll go interview at Pfizer, why not? The role seemed simpler than the others, where I basically just had to get bulk sequencing up and running, and they just wanted someone with bulk sequencing background. So it didn't even matter that I'd done single cell. And And ironically, all they needed was my grad school experience. So as my prior example, I would have been able to get into this company entirely from grad school, almost skipping my postdoc. But my postdoc gave me some additional sequencing experience that sort of made me a much stronger fit. And fit's really the idea that I want to drive home in this. Like, so the analogy here is you have a bunch of keys and a bunch of locks, and that we're kind of sorting to find the one that really fits you the best. And what'll happen is that as people are looking for all these different locks, you can feel like you're being rejected because none of these are willing to accept you. But in reality, it's really just finding a better fit. It's nothing personal. It's just, I'm looking for a trapezoid and you're a square, you know. And I think that taking that kind of larger view kind of helps also with ego management when you're nervous about these novel pursuits. So ultimately, I ended up accepting the role at Pfizer, and my interview there was quite surprising because, like, my entire career, I'd always been in this academic environment, and even MGH, where it was relatively more laid back, still had this air of like academic solemnity. But it wasn't until I went to my interview at Pfizer that I really got to see what true like security looks like, at least on a cultural level. Like, people were just laughing and joking. My phone screen, the hiring manager and her boss were just kind of laughing and joking on the line. I'd never even heard that before. Everyone else is always very like stiff and formal, and they're all just super laid back and chill and whatever. And just everything about the interview was like that. And I'm like, wow, this feels, it's like, it felt like a level of comfort I didn't know was possible. Like I exhaled and didn't know I was holding my breath. So that's what fit feels like, where you really kind of are in a space that's good for where you belong. And so the other thing is that entering industry for the first time, the sort of structure is very different from academia. Academia is very PI centric. You might have department heads, but it's not like as pyramidal as industrial personnel structures can be. So so at Pfizer, I ended up joining the my direct hiring manager, Angela Hadjipanayas, was the head of the NGS Technology Center, which is what we were sort of joining and building. That sat within the Molecular Systems Immunology Department, which was run by David Von Schack, which was a department within the Inflammation Immunology Research Unit, which was run by Michael Vincent, who's the Senior Vice President and Chief Scientific Officer. So you get this kind of aggressive org chart hierarchy that can go all the way up to the CEO. So Mike Vincent's boss was the president of all of R&D, and his boss was the CEO of all of Pfizer. So I was literally only 5 bosses away from the CEO of all of Pfizer, just to give you a sense of the hierarchy distance. And so this kind of brings me to what my industry job actually looked like once I got there. And so I came and helped build the Next Generation Sequencing Technology Center at Pfizer. And so this is somewhat of a unique model that resulted from Pfizer offloading their entire neuro department in 2018 when they were deciding to kind of step away and consolidate their expertise for only the areas that they have the biggest advantage in terms of expertise and resourcing. And so as a result, a lot of these equipment kind of was floating around, and they decided to centralize it. But they didn't want to just make like an agnostic core facility the way a lot of academic institutions do. Instead, they wanted to have PhD experts not only manage the equipment, but also stay on top of the new trends and help advise best practices to kind of create a center for technology rather than a core facility. So in a core facility, someone just hands you a sample. You don't need to know what's in it. You just process it. You spit out your FASTQ and then go on with your day. With the technology center, we had a workflow where we would work very closely with the biology leads, and not just in our own research unit. We would work across all 3 research units on the site. So the Kendall Square location not only has inflammation immunology, which is autoimmune diseases, but also had rare genetic disorders and metabolic diseases. And so I got to work on all 3 of those topics, and on any projects where people wanted to have a question that could be answered with NGS. So you might have your drug candidate you're testing, and you might have like a petri dish or a mouse model or a patient sample, and you treat that with your drug candidate or a control, and then you extract your RNA or your cells or whatever, and then you try to ask what genes go up and down differently between them, which is similar to a lot of the other NGS experiments I've done. So it's RNA-seq, bulk RNA-seq, some single-cell RNA-seq. And so the workflow arrangement we would have is we would meet with the biology leads and computational biologists. We design the NGS experiment with all the relevant experts in the room. We generate sequencer-ready libraries from the sample RNA and run on the sequencers. We'd collect and manage the metadata and upstream data processing, and then we'd hand it off to the comp biologists to meet again once the analysis is done. And so we would kind of also police people, make sure you label your tubes. How do we make sure that the metadata capture was done properly and all that. And then in the meantime, I could do development work. So I could test and develop new technologies. So we could get robots and play with automation. We could play with single cell surface marker barcodes. And eventually we ended up adopting an entire area of spatial transcriptomics, which is more or less a technology that does what my postdoc project was trying to do manually. That can kind of do all baked in. Another thing I was able to do is that because I was in the technology center, it actually put me in a unique position to provide expert guidance on up and coming technologies. So I was able to join a variety of consultation networks, and it was a great way to earn some like extra pocket money. So I could go out, meet with vendors, attend shows, demo instruments, participate in technology access programs, and stay up to date with the newest technologies as part of my job. But then I could turn around and sell that information to investors, inventors, and market analysts to provide a expert opinion on how well these proposed technologies are doing at meeting the needs of pharma. And so I'm in this unique position as a customer to provide customer feedback to these different stakeholders within these technologies. And then there's other things you can get paid to fill out surveys on spending habits. And I also had one thing where I had to go to like a focus group. They paid me to like come all the way out to like a corporate park and sit in some weird focus group where they literally asked me what color and shade of plastic my emotional response to as a potential like reagent container. So it's very weird, but I'm like, yeah, sure, why not? You pay me, I'll do it. And one of the nice utilities of this is that the consultation networks can provide anonymity for the clients and also compliance against disclosing material non-public information. So they're able to create a risk so that instead of you just wandering out in the wild yourself and being at risk of accidentally disclosing some corporate espionage, you can also have nice safeguards against that to kind of make sure that everyone's on the level. No one's asking you, so what are your secret drug targets? It's just like, what's your opinion as a customer of this technology that's providing genomic insights. So what ended up happening was in 2022, my boss took on a new role as the global director of data generation for precision oncology at Sanofi. And she was actually like, well, I mean, she's my current boss still, but she was definitely like the most intensive mentor I've ever had. All my other PIs were sort of very laid back, distant. You check in once in a while. This is the first time I had a boss or a manager who actually like just gave me straight up mentorship, walked me through every single step, like trained me directly, and also like taught me all of like the skills, taught me how to navigate corporate culture, taught me how to create checklists, taught me how to deal with the eccentricities of certain types of experts and egos, and just like thoroughly mentored and sponsored my development. So naturally, that's left a huge impression on my career and life. And so she ended up leaving the tech center in early 2022. And so I kind of took over as like a co-manager of the tech center with the head of outsourcing. And we kind of ran it for a while. But in the process, she kind of reached out to me as like, so are you happy? Just so you know, one of my FTEs opened up, and then that was it. And then I noticed a job posting popped up on LinkedIn. So I just applied normally and went in and ironically ended up giving a talk on the work I did my second year of my postdoc, which then which kind of created this interesting pattern of me now being hired twice on something I did 2 jobs ago. So I got hired into Pfizer largely based on work I did in my PhD, and I got hired in Sanofi based on work I did during my postdoc. So if the joke would be if I get a job next, it would be based on what I did at Pfizer. So so I ended up joining Sanofi in July of 2022. And so this provided me my first industry team lead position. So now, not only am I a new company, but I actually am leading a team of 3 other scientists in our genomics group. And so this is sort of a picture of our old and new building. So we started off in this kind of older building, and then last October, we moved into these 2 brand new beautiful buildings in Cambridge Crossing. So I'm kind of sitting on my 11th floor and enjoying my wonderful view from up there. So my current job today is quite interesting. One, the big reason why my boss and also myself were interested in coming here is that we get to work on cancer again. Like I did some cancer work in my postdoc and I liked it. And a lot of these new technologies that I was learning about were really tailor-made for cancer, especially spatial transcriptomics. And the opportunity to work on that again was really great. The other nice thing is that within precision oncology, we're allowed to work a lot more closely with the biology team leads. So at Pfizer, the biology team leads would kind of like do their own thing until they would have an NGS question. Then they would show up, we would help them answer the question, and then they'd go away again. Here, we actually get to be members of the project teams, where we actually get to sit in on the meetings that go all the way from like the chemistry and candidate design to candidate selection to PD biomarkers to safety to all the way into the clinic. And so we get a lot more exposure to the full drug pipeline here than we did at Pfizer. The other nice thing is that when Pfizer would go shopping for new companies with their COVID money, we would never see them. We would read about it in the news. Whereas Sanofi would go shopping for companies, we would get new coworkers and we would get new projects. And so like they get to go out and buy all of these bleeding edge technologies, and we could then work directly with the directors of those companies that are now internalized and get to play with these things. So you get that kind of small startup, high risk biotech experience without all of the other challenges that come with being at a small company, like being fired every 2 years or having it go under. Not to say there's a risk with larger companies, but you kind of get the best of both worlds here. I still get to like sit on project teams where they're trying to use these newer technologies, where we're trying to like make a cell stick to another cell directly using a variety of different means, or editing an immune cell to become a drug itself, or to take antibodies that are designed based on like llamas and alpacas and camels and using them as a way to encourage immune cells to attack tumor cells, and all that. And so what our team does is we can then work with the project team, and then I can go back to my team and we can then do bulk single cell and spatial readouts in support of those project teams as sort of a collaborative partner, rather than just kind of like a shop that they go to. And it feels more like a main quest than a side quest. And so I think that's sort of what's exciting about it. And so throughout my experiences, these are some of the things that I personally have observed as differences between industry and academia. Obviously, individual results may vary. But in my experience, industry seems to be more applied science. Every question has a clear and obvious utility for solving a problem in medicine. There's entire like domains of biology that we know about, but we just don't quite know how to approach things like, say, the 3D arrangement of genomes or the entire like jungle of non-coding RNAs. Like, there's biology there, it has an implication, but we don't have a clear way to like leverage that in a therapeutic way yet. But that is the space that all these brand new startups come from, and that's where the absolute tip of the spear exists. Another thing I noticed is that there's less stress around this publish or perish competition over this artificially scarce impact factor that journals like to perpetuate. It's not this big zero-sum game. Yes, there might be a limited number of strategic roles, but I've also noticed not everybody wants to necessarily be in a strategic role. There's actually a surprisingly large number of people who are very happy in pure execution roles. Another thing I noticed is that there's a more matrix style organization. Since there's access to more funding and expertise, it's much easier to just bring together these very large multidisciplinary teams that can accomplish in months what would have taken years to do in academia. And the other thing is that culture tends to be determined more by individual leaders. It seems to be that people are more often likely to quit bosses more than they quit companies. And in my case, I followed a boss rather than following a company. And obviously the pay can be significantly higher, at least at some of the mid levels, unless you get like some particularly prestigious academic posting. So that's certainly a good incentive. The other thing is that large companies tend to provide more stability, whereas small companies provide more opportunity. You're not going to like save the world at a large company, but there's a non-zero chance you could in a small company, because you come in at the bottom, you happen to be in a unicorn, you could potentially become like a millionaire, but it's like that's why we call them lottery tickets. You get paid in stock for a company that probably will fail, but you never know. and that networking is just as, if not more important than personal achievement. There are a lot of people who like to chase after awards and accolades, and those are all fine. But at the end of the day, it's really the networking that kind of creates the foundation for what's called career security instead of job security. You know, your company may go under, you may leave, like whatnot, but there's always like, you know, Boston is a hub. There's always another company down the street, and the networking is sort of what facilitates you having that landing pad in whatever your next company is. And that there seems to be a stronger focus on personal career development and goal setting, at least in the industrial places that I've worked, compared to the academic places I've worked, where a lot of the incentive seems to be, well, let's squeeze another year out of them because, you know, their labor is super useful. And there seems to be a somewhat conflict of interest between graduation and getting that extra paper out. And so this kind of brings me to some of the lessons that I learned throughout my career. The first and foremost is that rejection is not about personal inadequacy. It's about finding the best fit. And so my kind of personal motto is, if at first you don't succeed, raise your standards. Every rejection I've ever experienced ended up being a worse option by my own standards than where I ultimately landed. Yeah, I didn't get into exosome diagnostics, but that wasn't really a good fit because I would have just been sitting there pumping out libraries mindlessly than what I got to do at Pfizer. And so my attitude is, one of the hands worth two in the bush, you know, which is an idiom that just says, you know, the sure thing is worth more than the potential, which is usually true. But if you have the time, you might as well check the bush. You know, doing your due diligence increases your chances of finding the best fit. There are no stupid questions, only clarifying questions. This is something I would like to just like nail into the head of every professor I've ever witnessed bullying a student. This is absolutely unacceptable. And I think anytime a student witnesses that, they should absolutely push back. You should not be afraid to ask for pertinent information, no matter how much it may seem you're supposed to already know it. We have different methods of communication. We have different methods of learning. Yes, there's always another paper you could read. Yes, there's always another textbook you should know. But sometimes just saying it out loud says it, gets the communication happen. There's a reason why people just sit in meetings all day, because that's the most efficient way to get all this information across. We don't just tell everyone, just read this mountain of papers and get back to me. It's better to impress people who know you well than to impress strangers. Yes, accolades, awards, high impact factor status, and impressive CV will only ever get you as far as an interview, but positive working relationships will last your whole career. If you want to drastically increase your salary and job title, change companies. This is a pretty common situation. For whatever reason, people like to pay new hires more because of just the politics of it and the momentum of how you're known. So that's just kind of a rule at the moment with how things work. Maybe 20 or 30 years ago, it wasn't the case, but today it absolutely is, that if you jump around and change companies, you will increase your salary. Another warning that I need to warn a lot of my friends about is always talk to HR, business, or legal the same way you would talk to the police. Tread carefully and respectfully. I can't tell you how many times I've had friends get in trouble, even people in my own department get in trouble with HR, business, and legal, because they just naively bulldoze through things with only thinking about research and not realizing the implications with respect to the rest of the company. So that's just a little quick, you know, warning. And I could go on for another hour on that. And now that I am myself a boss, I've come to discover that reports can very often be more demanding than bosses. You know, people like to think that, oh, the boss gets all this fun thing, but with reports, you basically have to juggle them like you would students, and you have to be cognizant of their interests, their time, their needs, and their development. And so it's sort of an intensive mentoring experience as well, if done properly. And, you know, as I say, hard skills get you the job, but soft skills get you the promotion. A lot of people get confused because they think that they're very, very good at executing, but they're wondering why they're not then given a strategic role based on their execution skill. These are fundamentally different activities. And when you're in a leadership role, you are basically trading your execution in exchange for strategy, which means that you're kind of foregoing all the hard skills for soft skills. You use them to know what's happening, but you're not actually there doing it yourself as much. And finally, have fun. Science is a privilege and a playground. Explore and play. Like, I think people take this stuff way too seriously, and they forget how amazing it is that, like, the rest of society has decided to set aside money so that we can just ask questions and play with brand new technology and kind of be at that bleeding edge. And I think people get so caught up in the politics and the minutia that they stop to just look around and marvel at how incredible this all is. You know, and that was the first lecture I got when I joined UConn. that science is a privilege and we take it for granted. Like, and there's no reason the public couldn't just decide, no, you know what, we don't want to fund NIH or NSF anymore. Like we'd rather have like healing oils or something else, and they could do that. Like, so don't take it for granted. So that kind of brings close my story. So I'm happy to answer any questions, and also feel free to reach out to me on LinkedIn. My rule is basically, if you just send me a message, hi, my name is, this is my thing, I will happily connect with you. I don't, I'm happy with cold introductions. I had a grad student from Vermont randomly reach out to me, and I gave him a full warm introduction or a full informational interview with a flat out cold introduction. But some people like to just spam random names without a message, and that kind of fills up your inbox. But literally just a single, hi, my name is, this is my situation, I'd like to talk to you, would be great. So.
Fantastic. Thank you so much. I really liked those lessons learned at the end. I think about my own journey and things that I wish I could tell my past self. So I guess I'll ask the first question, which would be, if you could tell your past self any one thing to help you get through what you were going through, what would that be?
Ask for help. That's it. Just ask for help anywhere, anyone, everyone. That's it.
I think a lot of times we get so stubborn in that we feel like we have to do it on our own. And if we don't ask for help, if we ask for help, we're failures. And so
well, in my case, I was afraid of being judged. And I would say to my past self, yes, you'll be judged. They're wrong. Do it anyway. Like, ask for help. Like, if they're not going to help you, move on to the next one.
Excellent. We had another question come through, which would be, if you had to do it over all again, would you still take an academic postdoc out of your PhD?
Yes, because I got a chance to get settled into the hub itself. What my postdoc offered me was less about like the hard academic and more about like, well, I just threw this stuff on the sequencer. I've got like free time. I'm going to wander around and just attend like classes or, pester the guy over at the Harvard, high, what's the word, like the supercomputer that they use for data processing, and just kind of like it gave me that 2-year acclimation and space to actually just kind of wander around and learn my way around Boston itself.
Fantastic advice. We have another question coming in. How did you overcome or manage your imposter syndrome to continue your PhD journey?
That was what the meetup was for. So by redirecting my attention to something where I was strong, I was able to rebuild my confidence.
I think that's important. I think all of us should realize that we should do other things other than just science, right? We're more than just scientists. And I tell my students that now. I played music when I was in grad school, and my students now, they're like, wait, why do you play music? What do you expect? I go home and play with test tubes in my apartment? Which I'm like, I wish I had test tubes in my apartment, but I think it's important to broaden your own experiences. Another question, how did you navigate your interpersonal relationship with your PI or lab members when things weren't so good?
I hid. I wouldn't say I did that well. So in grad school, I basically hid and avoided it. And my PI was always just like, oh, shucks. Like every time I'd be like, okay, this isn't working. Oh, shucks, try again. Like wasn't a lot of like aggressive thing, because it's just like I'm at the mercy of bacteria functionally. Like, okay, I put all the ingredients in the tube, I hit go, I get no colonies again. Like, well, what are you going to say to that? You just, this is the only button we have, and that's that. And you just go until you don't. And when my postdoc, when I went to talk to my PI, he was already trying to persuade me at the very same thing I was trying to argue him for. So that was a different situation. I would say that was a mistake I made. In retrospect, I should have wandered around the institution to find a PI who was interested in actually mentoring, and just go until you find him. Like, or even outside the institution, like why just email somebody? There's other experts. Like, who's your PI talk to in their community on the same subject? Like, I mean, there's also things like Reddit, you can go to like Lab Rats or something, like just wherever, just anywhere and anywhere.
Yeah, another question coming in. Would you have rather done an industry postdoc as opposed to an academic postdoc if given the chance?
Yes, I would have. However, I think they're different things. If I'd done an industry postdoc, I probably would not have been as proactive about like getting out into the city the way I did with my academic postdoc, because industry, I would have just been fixated on trying to like have a landing place. It would have been a smoother transition, but it would have been less diverse.
Excellent. We have somebody asking, could you show us how to ask for a new warm introductions from your network?
Yeah, I'll do it right now. Hi, my name is Seth Garren. I currently work at Sanofi. I noticed in your profile that you have such and such a thing that I'm interested in. I was wondering if you'd be willing to spend some time to talk to me more about that.
Perfect. We also have resources for like that, just to, talk about our own, our own resource. Beyond the PhD has stuff on like how to use your LinkedIn properly. So definitely go take a look at those resources. But that's exactly what we're doing. That's how I got in contact with Seth. I reached out. I noticed that we both graduated from UConn. I'm always looking for people to come and talk. I said, hey, we both graduated from UConn. I would love to know if you want to share your story to Beyond the PhD. And well, here we are.
Well, what's funny is you reached out to me like right as I was having a networking meeting. So like one of my contacts who I met at a networking event, who had actually applied to join the team I was in at Pfizer, but ended up leaving because it wasn't quite high enough to her standards, just reached out to me randomly, had a networking meeting. And then right as I left that, you messaged me. And so it's just like right even the timing of that.
Yeah. The importance of networking is really so important. I think that question's kind of coming through here. You had mentioned weak connections. How do you maintain those relationships? Or is it even important to maintain relationships with those weak connections?
I mean, if you can get a cold connection to respond, then a weak connection is easier to respond. And one of the things I also learned, this sounds super cliche, but it is for a reason. There's the book, How to Win Friends and Influence People. And the core message of that book is the favorite topic of literally everyone is themselves. So if you ask a random person to talk to you about their favorite topic, they will like you. And if you take sincere, genuine interest in it, they will like you even more. So it's kind of a natural fit if somebody's already an expert in something that you're interested in. Like, I like all of you because you're taking all this interest in my personal life. You're all really cool. So I think it seems intimidating because you're like, oh, there's this person who's older than me and more credentialed than me. They're not going to do it. But it's you just spam people on LinkedIn and you're definitely going to get a bite. Because I think people are mostly flattered if some random grad student is like, hey, I saw your profile. I was wondering if you would be willing to take some time to just chat with me sometime. I think everyone I've met would be very flattered if they receive that message. So you can do that for a stranger. You can do that for a weak connection. Hey, I don't know if you remember me. We met at that Biotech Tuesday 4 years ago. Anyway, I saw this market. Like, that's literally the person I just had the meeting with when you messaged me. Was somebody who's like, hey, I honestly didn't even remember her from the event, but I'm like, sure, I will do everything I can to help you get this job. And turned out we weren't quite up to her snuff. So ironically, she rejected us anyway. So
yeah, it's always worth a shot. Worst case scenario is they say no.
Yeah, or more often than not, they just are too busy and don't respond. But
yeah,
I mean, the thing I don't like is when people just add me and say absolutely nothing, because then I'm like, okay, who are you? Who do you know? Have I met you? Are you just a recruiter just filling a network? Or are you somebody who I just forgot about? So like, if you send me anything, you're just like, hi, my name is, I'd like to talk to you. That puts you in the top 1% of people who've sent me connection requests.
Perfect. Any other questions? I'm scrolling through the chat. I think I covered everyone. It looks like no. As Seth said, he's more than welcome to connect. Just, do message and say, hey, I attended your seminar that you offered with Beyond the PhD back in January. I'm sure that will work. I do want to thank you again, Seth, for taking some of your time to talk with us today. I hope that everyone here, if you're new to the Beyond the PhD community, follow us on beyondthephd.com. Check us out on Facebook, Twitter, LinkedIn and Instagram. There we have all sorts of content that you could follow. Join our mailing list, find out about upcoming seminars and workshops and all that sort of fun stuff. And I hope you all have a wonderful 2023 if I don't personally see you at any other point. So thank you again, Seth, for joining us.
Thank you.